Hello and welcome. We will continue our discussion on biophotonics, and today I have for you a very exciting and very new and very promising new field of light activated therapy. Remember that one of the two major goals of biophotonics is curing of human ailments. Thus far we have seen how light could be used for imaging beyond diffraction limit, imaging at a very small scale, imaging just few cells or intracellular components. At the same time, we have seen that how light could be used for sensing or detection. We are able to detect single molecules, single viruses, or different types of pathogens, or early detection of cancer using optical biosensors. So, light has thus far, as I have told you, we have seen is being used to both image as well as to detect and sense diseases, pathogens, things that cause diseases, etc. This time, in this particular module, we are trying to see if we could use light for curing any such disease that we have either imaged or we have sensed or detected. And that could be, there could be a plethora of different types of diseases, both communicable as well as non-communicable, something that has been caused by an external agent like a pathogen, or something that has triggered in your own body. Say, for example, some kind of gene mutation have taken place, as a result it has caused cancer or some sort of an autoimmune disease. So, we are going to discuss this new topic and this very interesting exciting topic of light activated therapy, phototherapy, and in this particular case we are going to discuss about photodynamic therapy, how to utilize light to cure certain specific diseases. So, let us get on to it. What exactly is photodynamic therapy? So, photodynamic therapy is a treatment that involves very simply, PDT, photodynamic therapy, is a treatment that involves light sensitive medicine and a light source to destroy abnormal cells. Okay? You produce or you inject into the body some sort of a medicine that is sensitive to light. You excite that specific medicine with light, with specific bunch of photons, with specific wavelength, with specific energy of photon. That light activates the medicine, and then the medicines perform some sort of a function, a biochemical reaction, say for example. Upon activation by light, this medicine, this drug, this chemical agent performs certain chemical reactions, biochemical reaction, and that in turn destroy abnormal cells. So, these are two of the major free picture that I could get. Usually the light source is laser and you are delivering it into the human body. This is actually a keyhole type surgery. Person's body, a certain section of the person's body has been opened up. You could see the scalpel and scissors, and thereby they are inserting a tube which is basically a optical fiber, and that is containing the red laser light, and that red laser light is used to damage, destroy, annihilate abnormal cells. And this therapy, this photodynamic therapy, is approved by the Food and Drug Administration, FDA of the United States. This is one of the largest drug administration company, drug administration agency, not company, it is not a private company, it is a federal government's branch, federal government's agency of the United States, and it is actually a very big deal to get FDA approval. Several of the vaccines that you are seeing these days going around for coronavirus detection, as well coronavirus prevention, as well as several other types of vaccines and drugs, they need to get FDA approval if they want to sell their product in United States or territories that are under the jurisdiction of United States. And it is usually the case that they are one of the stringent agencies, and if you are able to get it passed through them, it is easier for other agency to approve as well. Not always the case, not always the case, there are exceptions, but this is a bona fide, the point that I am making is this is a bona fide approved therapy for treatment of cancers. You know about chemotherapy. I told you in the previous classes, the introductory classes, that I will be taking phototherapy or light activated therapy, and this is the one which it is. This is photodynamic therapy approved by one of the highest agencies of the United States government, and we are seeing more and more use of PDT, photodynamic therapy, in treatment of cancer. So, what exactly does PDT do? What is PDT in a more detailed way? Let us try to understand that. So, as I said, photodynamic therapy is a treatment that involves light sensitive medicine and a light source that destroys abnormal cells very easily, and it is used to treat some amount of skin and eye condition as well as certain types of cancers. It is, though heavily used for the treatment of cancer, you will see that it is being used for other such conditions as well. Including it has found applications in ophthalmology, dentistry as well as dermatology. On their own, the medicine and light sources are harmless, but when the medicine is exposed to light, it activates and causes a reaction that damages nearby cells. Again, the medicine is light sensitive. The medicine will only work when it has been excited by a specific wavelength of light or group of specific wavelengths of light. Maybe one, maybe more than one, but there is a specificity of it. By itself, the laser is harmless, the medicine is harmless. You combine them two, the medicine then starts producing some kind of a biochemical reaction with the surrounding area, and that this surrounding area, this biochemical reaction that this medicine causes, ultimately destroys the surrounding areas, which happens to be either the cancer cell, the tumor, the malignancy, or something else that where the medicine has been injected. It allows small abnormal areas of tissues to be treated without the need of surgery, and this is the most important part. It does not require surgery, and it has been used to treat abnormal cells in different parts of the body such as skin, eye, mouth, food pipe, esophagus and lungs. And this is also approved by the National Health Service of the United Kingdom. This is the governmental health service, NHS. I myself am a beneficiary of NHS. This is a fantastic service, and NHS is the government of United Kingdom's, government of Great Britain and United Kingdom's health service, almost free medical health service almost, and it also utilizes it. So, I am not telling you something that is coming up in the future. Yes, it is coming up in the future, but certain section of it is already regularized, already optimized, already approved by various health organization, various health approval agencies all over the world. And one of the major important advantages of phototherapy, light induced therapy, is that the tissue can be treated, the disease can be treated without the need for surgery. Now, the medical students listening here will give you a whole plethora of reason why several times surgery is not that beneficial. You are opening up a tissue and you are producing some sort of incision. Though here as well, like in the previous slide, I told you that incision could be done, but on several cases PDT can avoid surgery altogether. PDT can avoid surgery altogether, and there are several areas where cancer may form which is unoperable. like say there is a tumor somewhere in the middle of your brain. It is very, very difficult to take it apart, take up the cranium, take up the apart the skull, and taking your scalpels and scissors and whatnot inside the brain and taking that tumor out without damaging the other parts of the brain and thereby risking permanent paralysis or even death. So, there are several areas where cancer can form which is simply inoperable, which cannot be touched by surgery. PDT has the potential, mark my word, has the potential to go into those areas. Light could be shined either externally or by making some kind of a keyhole, not exactly going to that, but the light can be made to penetrate through the tissue and thereby activating the medicine that it has that has attached to the tumor, and then causing the biochemical reaction. The biochemical reaction in turn destroy the tumor. So, that is the overall very rudimentary, very basic principle without getting into too much medical jargons. I mean, when I was doing my own study, my own research on PDT, I had to take a mini course on cancer, and believe you me, that is incredibly rich as well as complex field, and hats off to those medical students and scientists who spend their lifetime studying cancer and oncology and becoming specialist in oncology. This is truly a fascinating and very, very complicated subject. We, the people who are staying outside, are hardly scratching the surface. What is carcinoma, what is melanoma, these are the very basic things, but then how complicated it goes and how heterogeneously the cancer spreads, all of those things are quite complex and quite fascinating, and I thank all of the oncologists whosoever is listening for your work. So, let's move forward. What are the different specific conditions that are treated by photodynamic therapy? Well, you can read several of them. I am particularly interested in macular degeneration. Macula is a specific part of your, usually central part of your retina, and as a person becomes old, this macular region simply degenerate. A part of your retina simply degenerate, resulting in severe amount of vision loss. You do not usually get completely blind, but partial blindness can be caused. So, and we do not know what is the main reason triggering macular degeneration. You must have heard several old people after age, reaching a certain age, they are losing their eyesight, not necessarily always because of cataract or because of some kind of eye injury. There is something significantly called macular degeneration. It's a part of your retina simply degenerate. A part of your retina simply refuses to take light, and there is a degeneration forming, and because of that your vision is partially lost. Then there are other types of various types of cancer. Basal cell carcinoma, you have Barrett's esophagus, where there is a change of cell lining in your lower food pipe. Mouth cancer, oral cancer, as I said, technically oral cancer is an Indian cancer. Oral cancer is mostly prevalent in Indian subcontinent area, simply because we chew tobacco. This is our lifestyle. We chew tobacco rather than smoke tobacco. In Europe and North America, those people who smoke tobacco, they develop the risk of having lung cancer. But chewing tobacco, betel nut or different types of those pan masala, etc., beg your pardon, causes mouth cancer. Oral cancer per se, mouth cancer is the overall set, oral cancer is a subset, and they can be treated using photodynamic therapy without the requirement of doing a full-fledged surgery and without doing a specific chemotherapy. This is phototherapy. This is phototherapy. Light is being sent to destroy the abnormal cancer cells. In chemotherapy, a drug is specifically targeted to destroy highly replicating, highly dividing, highly multiplying cancer cells. Here, it is the light that is being generated, the light is causing some kind of a biochemical reaction that is in turn destroying it. It has also been used for warts, acne and other extramammary Paget's disease. These are several precancerous conditions, various skin type conditions, and my source here is National Health Service, United Kingdom. I am not randomly taking these names out and saying that PDT could be source. These are actual things, these are actual diseases, specific diseases that have been treated, that is already been treated, and the complete procedure exists for light based therapy, for photodynamic therapy of all of these. And you will look, most of them take care of the skin cancer part. Skin cancer, to the best of my knowledge, is little less prevalent in India. We mostly have mouth cancer, breast cancer, etc., but they are the one of the first to utilize PDT. So, what are the procedures? What exactly you do in photodynamic therapy? What exactly is done? The patient comes and the patient is injected a specific drug. This drug is the PDT drug. This drug is the one which gets activated. This is the medicine, this is the drug that will only get activated upon exposure to light. This drug will only get activated upon exposure to light. So, after the drug has been injected into the patient's body, the patient need to wait for something around 24 to 72 hours, it depends on the drug, and the drug gets accumulated in the tumorous region. The drug gets accumulated specifically at the area which is supposedly abnormal, which is showing metastasis, which has formed a tumor which we need to treat. So, the drug specifically get accumulated at the cancerous growth, at the growth which is causing problem, which is the basically the disease forming. So, this drug after 72 to 24 to 48 and 72 hours get accumulated at a specific location within your body where you have the tumor. Then we induce light into that particular area. So, this is the zoomed in version, this is the zoomed in version of the area, this is the zoomed in tissue, these are the normal tissue, this is the overall tumor that has formed, this is the overall tumor that has formed, this is the cancerous growth, this is the metastasizing part, and you shine light onto it. The photons create some sort of a biochemical reaction of the drug which is attaching itself selectively. By this time you know how we can attach specific biomolecules to specific location. Remember, we discussed this in the previous class. We do mostly antibody antigen complexes, these are very specific. There are other processes, aptamers are coming up very strongly, some sort of electrostatic procedures or some kind of thiolation. These are the way in which a specific drug could be attached to very specific location of a particular region of a cell or a tissue. So, your drug has mostly been accumulating in the cancerous growth. You shine light, the light creates some sort of a biochemical reaction, the light induces a biochemical reaction onto the tissue, and the tissue therefore gets destroyed, the tissue therefore gets annihilated, the tissue therefore gets burnt, dissolved, vaporized or simply annihilated, leaving the nearby cells which the drug has not accumulated into with minimal damage. Please put an underline over the term minimal damage. Minimal damage does not mean that there is no damage, minimal damage. So, this is a very, very localized therapy. This is very, very localized therapy. Ideally, only the cancer cell is destroyed, only the cancerous growth, only the group of cancerous cells, only the cancerous tissue is destroyed, leaving nearby areas, nearby surrounding areas healthy or untouched or minimally damaged. That is the overall basic principle of your photodynamic therapy. Now, again, I am just repeating myself, the whatever I showed in the cartoon in previous step. This is something which I have written down because many of you in the feedback asked for notes. I have previously just given you the cartoon and have spoken to you about it, but many of you are complaining that you need detailed description, detailed notes, otherwise you are having difficulty preparing for exam, and that is all right. So, I have written what I previously in the previous slide I just mentioned, that you administered the PDT drug. There is a longer retention of the PDT drug by the malignant tissue. Malignant tissue is the cancerous tissue, the cancerous growth. Delivery of light, generally laser light into malignant tissue site, either by entering through a fiber, or if it is on a skin area, you can externally shine light onto it. If it is some sort of an orifice, if it is reachable through orifice, you just insert, or if it is somewhere else, you make some sort of a keyhole surgery and then insert the light. Remember, if this is the tissue, light has to reach this place. It does not mean the fiber has to reach this place. The fiber can be few millimeter, few centimeter away, as long as the light, just like your torch light, you switch it on and the light emanates out of it. So, even if your fiber is quite far away from it, if you have made sufficiently high intense light and you have ensured that this light is not scattered or not too much absorbed by the surrounding tissue, you have chosen a specific wavelength, a specific energy, specific wavelength, specific intensity, it can penetrate through, or some amount or substantial amount of it can penetrate through, some amount will always be absorbed or scattered. It can go through and activate the medicine that is in that cancerous growth area, thereby causing a chemical reaction and destroying it. So, the fiber does not need to be directly in contact with the tumor. The fiber does not need to be directly in contact with the tumor. The fiber could be far away. It could be like a lamp, a source of light. It could be like a lamp, like a light bulb, like a torch light, something of that sort, that as long as somehow the light, the photon, the energy is able to reach the tumor site which contains the medicine, right. That is the overall thing. And what is the biochemical reaction? So, for all these slides I have been harping about a biochemical reaction is forming, a biochemical reaction is forming. What is the biochemical reaction? It creates highly reactive oxygen species. The biochemical reaction is formation of ROS, reactive oxygen species, oxygenated singlet or triplet oxygen states, singlet or triplet oxygen molecules. These molecules are highly reactive. Chemistry students call them radicals. You should know, irrespective of whether you are a student of chemistry or not, you should know what radicals or free radicals are. You should know. I am not going to define you what free radicals are. They are oxygen radicals, they are reactive oxygen species, they are highly reactive oxygen molecules, and they basically oxidizes the surrounding area being the tumor in which the medicine is present. So, you have a medicine which is attached to the tumor, you are shining light onto the medicine is creating oxygen species, this oxygen species is oxidizing the surrounding area. What does oxidation means? Burning. Oxidation simply means burning. It is burning its surrounding areas. The surrounding areas are the tumorous cells, and since it is a very localized hit, since a particular dose have been optimized, a particular amount of drug is present there, it is producing a specific amount of oxygen species depending on how big or how small your tumor is. Based on that, you generate, you destroy the localized cell, you destroy the tumor cell without harming any other cell in the nearby area. Basically, you optimize, you calculate how much amount of drug can produce how much amount of oxygenated species. This much amount of oxygen species is required to destroy these many number of cells. You make a calculation of how many cells have actually shown malignancy, how much metastasis is there, i.e., what is the size, how much cancer has spread, how many cells you need to destroy. That's basically the overall thing. And from a chemical reaction point of view, this is the overall chemical reaction. Now, I tried to figure out, but these are the usual notations used. The PDT drug, which is considered P after light absorption, goes to a singlet state. Remember, we discussed singlet and triplet state while discussing fluorescence and phosphorescence in Jablonski diagram in, I think, lecture number 5 or lecture number 6, where we have used the notation S1 and T1, singlet and triplet state. S0 is the ground state, paired. S1 is paired excited state, and T1 is the unpaired triplet state. I do not know why, but the pharmacy people, instead of writing S1 and T1, maybe it overlaps with something else they donate, they use the singlet state as 1 P star and the triplet state as 3 P star. So, this is the drug. When it becomes S1, they put a star which probably shows excitement, and 1 stand for singlet, single is 1, and triplet stands for 3. I kind of like this one more than S1 and T1, but it is the same thing. So, you should not get confused. This is the singlet state. This 1 P star is equal to S1 and 3 P star is equal to T1. This is paired, this is unpaired, paired excited, unpaired excited. This shows phosphorescence, this shows what again? I forgot, you have to tell me what it shows. So, I am not going to tell you singlet and triplet once again. So, the PDT drug upon light absorption becomes singlet state, excited singlet state. It goes into intersystem crossing and it forms the triplet state. This triplet state reacts with either the hydrogen or the oxygen already present. These are the two most common thing anywhere in biological cells, biological tissue, biological matter. Some amount of moisture, some amount of humidity, some amount of water content will always be there if we are talking about biology of this specific planet. Yes, there are exobiology which deals with biology of extra other planets. So, no, I am not making fun of you. So, you will find oxygen and hydrogen common in most biological matter. These triplet states react with this oxygen or hydrogen, produce highly reactive, this dot chemistry student knows are radicals, produces highly reactive radicals, reactive oxygen species. They basically oxidize or burn the surrounding tissue area, oxidation of cellular components. They oxidized certain areas of the cell, and you will see that not entire cell needs to be destroyed, destroy some of its major components, destroy the nucleus, destroy the mitochondria, destroy the cell wall or the cell membrane, the plasma membrane, the cell will undergo destruction, right. You do not have to completely annihilate every single part of the cell. The major important part, destroy the nucleus, the cell will die. Destroy the mitochondria, the energy part has been removed. So, certain parts, the oxidation of cellular components, certain components are burnt by this reactive oxygen species, resulting in cell death. So, these are the free radicals. These are the free radicals that will impinge upon certain cellular components, mitochondria, cell membrane, etc., destroy it, and the destruction of one such vital cell organelles will result in destruction of the entire cell, even if the cell is tumorous, even if the cell has undergone metastasis, even if the cell is showing mutation, even if the cell is cancerous and what not. You destroy certain part of it and it simply falls out. So, what is this drug? This light induced medicine, PDT drug, this will only go into these different states upon light absorption. They are called photosensitizer. They are called photosensitizer. A photosensitizer is a molecule or drug that produces a chemical change in another molecule in a photochemical process. The suitable photosensitized PDT should be. So, this is a light activated or light sensitive molecule which can undergo. So, PDT is the overall molecule, a part of PDT is photosensitizer. Just like you remember chromophore we were discussing when we were discussing our eye, not the entire molecule need to be excited by light. You have fluorophore, you have chromophore. So, photosensitizer is that part of the PDT drug that activates, that gets excited by light and that activates, that brings in the overall change in its singlet and triplet states. Just like your chromophore, remember from the discussion we had about vision, how we see the chromophore, cis retinal, etc., how it is getting excited by light and a chain reaction is forming, is the same thing. The photosensitizer is the part, is the specific part of the PDT drug that produces a chemical change, that induces a chemical change, i.e., it induces the entire molecule to go from excited singlet state to excited triplet state, and then this triplet state, states react with oxygen or water, producing radicals or producing reactive oxygen species. And here is the drug development part. Here is the most important part, that this photosensitizer must have the ability to selectively accumulate it into cancerous and precancerous tissue. The efficiency of this drug depends on its selectivity. It cannot simply spread all over your body and destroy everything that it has touched or it has got in touch by shining light. It has to be very, very, very selective. So, think about how everything is coming into full circle. You do a spectroscopy first. You do a spectroscopy first and figure out what are the molecular changes between a normal cell and a cancerous cell. What molecules have changed? It has to undergo some sort of a molecular change, some sort of mutation has taken place, thereby either the protein has misfolded or there is in the DNA base pair something has changed. Instead of AT, you have ATCG, adenine, thymine, cytosine and guanine. Some sort of base pair changes have happened, resulting in some kind of molecular change from a normal cell to an abnormal cell to a cancerous cell. You understand the abnormality. From that abnormality, you develop a drug that can only attach to with that abnormality region. And that is a huge area of active research. Not everything that we have, not every PDT drug is selective. The efficiency and overall the cost also depends on how selective it is, so that it accumulate only in the cancerous and the precancerous tissue. It must target specific cancerous cell, and this is the point that I figured out. Cancer does not spread homogeneously. It does not spread. So, even if it is one area where the cancer is spreading, say for example, mouth cancer or oral cancer that is spreading, it is spreading at a different state. The same tissue, different areas of the same tissue will can show different stages of cancer. Somewhere in the same oral tissue, 1 centimeter by 1 centimeter area, say 1 millimeter by 1 millimeter area, you see different spread of cancer. So, it has to selectively accumulate to those areas where cancer is present and some amount of precancerous first early stage of cancer has been present, and that is the one of the most difficult part, how to target that, because the earlier precancerous you become, the difference between normal cell and a precancerous cell is very less. Maybe you are able to detect it using some kind of an optical biosensor. You detect it, figure it out, and then you target it using some kind of molecule. Do you see how everything is connecting together? The photosynthesizer should be amphiphilic, so that it has a hydrophobic part, it can go inside cells, as well as it should be water soluble, so partially water soluble, so that it can spread throughout the body. It should absorb significantly at wavelengths. These are the wavelengths that has maximum transparency for biological tissue. I think if I have not mentioned it earlier, this is the window of transparency for biological tissue. Biological tissue mostly starts absorbing or scattering at longer wavelength, mid infrared. I have mentioned that, and this allows more amount of photons to simply penetrate, and it should exhibit minimum toxicity as dark, meaning you have not shown the light. If you have not shown the light, the drug should not work. That is the point. Okay, that is the point. What is the point of having photodynamic therapy if your drug does not require light and simply start killing or oxidizing anything that it comes in touch with? High quantum yield should not aggregate, and most importantly, once the process is finished, i.e., when you have oxidized the metastasis tissue, when you have destroyed it, the photosensitizer, whatever is remaining of the drug, should be rapidly excreted from the body, right. So, you have a drug that got accumulated into the tumor destroyed, whatever residue is left should be excreted out, of either renally discharged or fecally discharged. It should be simply removed from your body. That is the type of material that we are looking for. One of the earliest such a drug is this porphyrin, hematoporphyrin. This is the drug. If any pharmacy students watching this, I want to give a shout out to you as well. You guys are also awesome. They spend their lifetime, developing these sorts of drugs which have these either some sort of a functionalized group which gets attached to specific tumor sites. Think how difficult the process is and how much accuracy they require. This should only be attaching to a specific area of the tumor cell without affecting others. This is one such example, hematoporphyrin. Porphyrin is a very common drug used in pharmacy. I think most medical students must have heard, most life science students should have heard of porphyrin. I have heard of porphyrin. So, one of the first group of photosensitizer that was approved by FDA, it gets excited by 630 nanometer light. So, this was your window of opportunity. 630 is obviously falling below it. So, it is obviously, since it is the first generation, so efficiency is less, only 1 to 2 millimeter penetration depth. And the problem with porphyrin derivatives, the earliest, the generation 1 of these kinds of PDT drugs were unsuitable for tumors which are more than 5 millimeter from the source of illumination, and patients were asked to avoid direct sunlight and bright artificial light for a period of 4 to 6 weeks. After you have been treated, you have to stay at home in darkness for 4 to 6 weeks. Otherwise, the residue that is remaining into areas which are non-cancerous could be oxidized as well. Since then, life has moved further. You have developed all of these different types of drugs. This is obviously not an exhaustive list. Lutex is coming up very, very strongly. This follows in that range, 530 to 770 nanometer. It is already proven against breast cancer and malignant melanomas. Melanoma is skin cancer, a type of skin cancer. Oncologist will go into my throat for oversimplifying it. Non-melanoma skin cancers are verteporfin. I cannot pronounce them properly. So, these are basically the area where they absorb light and thereby produces reactive oxygen species. So, this brings me to the end of the topic today. I simply gave you a brief introduction of what PDT is. the principles, the mechanism, and a little bit of photosensitizer. Photosensitizer is the photosensitive molecule which forms part of the overall PDT drug. The PDT drug basically upon activation by light produces reactive oxygen species. The reactive oxygen species oxidizes, i.e. burns the nearby surrounding areas. So, please visit the National Health Service of United Kingdom. They have a fantastic website describing the various different human ailments in a very simplistic term. So, more than going into Wikipedia, I go into NHS website if I have to understand disease, and since they are the governmental website of United Kingdom, they have mostly authenticated data. Wikipedia can be unauthenticated, anyone can edit it, not National Health Service. National Health Service is the standard version. If I have to look for something medically relevant and standard, either is National Institute of Health, NIH, United States, or NHS, which is United Kingdom. So, thank you. I will continue with the discussion further. Thank you.